This video is about the food tests that you should know for your IGCSE Biology exam. Food samples can be tested for the presence of the following biological molecules: starch, reducing sugars, fats, proteins, and vitamin C. Iodine solution test for starch. Starch is present in foods like potatoes, rice, bread, pasta, etc. So simply we can use the food in the solid form. We have to place a sample of the food on a white color tile. The idea is to see the color change clearly. Then add a drop or two of iodine solution onto the food using a dropper pipette. If starch is present, iodine solution would change from brown to blue-black color. If starch is absent in the food, the iodine solution will remain as brown color. Benedict solution test for reducing sugars. All monosaccharides and some disaccharides such as maltose are reducing sugars. This means that they will react with the Benedict solution, which is light blue in color. If the food sample is in solid form, first we have to cut or grind the sample of food and place it in a test tube and add some water and mix very well so that the sugars will dissolve in water. So from that solution we can take 2 milliliters and transfer it to another test tube and then add another 2 milliliters of the Benedict's reagent. Then this mixture should be heated in a water bath. Boiling is not necessary. The heating to about 80 degrees Celsius is enough. When the mixture is heated in the water bath, if reducing sugars are present, we can see a color change. First, you can see the light blue color converts into a green color. Then gradually it will be turned into yellow, orange, and finally a brick red color precipitate would be formed. So a brick red color precipitate would be formed if large amount of reducing sugars are present in the food sample. If only a small amount of sugar is present, a green or yellow precipitate will be formed. If there is no reducing sugar, the Benedict solution remains light blue. This picture shows the results of the Benedict's test for different carbohydrates. Lactose contains reducing sugar because you can see a brick red color precipitate. When the test is done for starch, the Benedict solution remains blue because starch does not contain any reducing sugars. Since glucose is a reducing sugar, you can see a red color precipitate is formed. Fructose also has given a red color precipitate. Sucrose is not a reducing sugar, so the blue color of the Benedict's reagent remains there. Testing for fats and oils. This test is called ethanol emulsion test because we expect a white color emulsion to be formed if fat is present. So we know that fats are insoluble in water, but they dissolve in ethanol. So the first step is to chop the food into small pieces and shake it up with ethanol. What we expect here is if there is any fat, it will dissolve in this ethanol. Then we can separate a clear solution from this mixture and take another test tube with a little bit of water, about 2 milliliters, and then we can add the ethanol mixture into the test tube containing water. If there was any fat in the food, we can see a white color emulsion is formed. You can clearly see the white color emulsion, how it looks like in the diagram. This is because the fat ethanol mixture breaks up into millions of tiny droplets. So this white color opaque milk-like mixture is called an emulsion. If there was no fat in the food, the mixture of water and ethanol remains transparent. Testing for proteins. This test is called Biuret test. Here the food sample should be in the liquid form. To the food sample, you can add some potassium hydroxide solution and a few drops of copper sulfate solution. So instead of using these solutions separately, we can use biuret ready mix reagent, which is already available in the lab. So once the solutions are mixed, you have to shake the test tube gently. If proteins are present, it will turn into a purple color solution. If proteins are not present, it will remain light blue in color. Since potassium hydroxide is a strong alkali, always we have to wear eye protection when you perform this activity. This diagram shows how biuret test is performed. We have to mix the food sample with copper sulfate solution and potassium hydroxide solution. If proteins are present, it will give a purple color. If proteins are absent, it will remain light blue color. Testing for vitamin C. Vitamin C is present in most of the fruits and vegetables. The sour taste that we get from most of the fruits is due to the presence of vitamin C. So vitamin C is not a carbohydrate, not a fat or protein. A blue color solution called DCPIP solution can be used to test for the presence of vitamin C. DCPIP loses its color or decolorize when vitamin C is mixed with it. So into a test tube, we can put about 1 milliliter of DCPIP solution, and then using a dropper pipette, gradually we can add the food sample into the test tube with DCPIP. If vitamin C is present, blue color solution becomes colorless. If vitamin C is not present, it will remain its blue color.